What if it was possible to treat incurable diseases such as Huntington's disease, HIV, or even cancer? Or what if it was even possible to help prevent unborn babies from developing these inherited diseases? Well, this all may be made possible by something so small it cannot be seen by the naked eye. Now, what if I told you that something was in the very thing that can make us sick? That's right. This almost universal answer to all of these problems lies within bacteria. This breakthrough tool that was discovered in bacteria was something that was already naturally occurring. Like you and me, humans have immune systems that attack and destroy foreign invaders that make us sick. Bacteria have immune systems too, but scientists discovered something interesting about them. There was a part of their DNA that was involved in their immune system, or more specifically, the defense against viruses. The problem with these viruses is that they need a host in order to survive, so that they can take control of the bacteria's cellular machinery in order to replicate. This DNA segment of interest, aka Crispr, was originally discovered in the commonly known bacteria E. coli. Scientists noticed that it was composed of short repeating base sequences that were separated by something known as spacer DNA. This spacer DNA is the identifier of these viruses, as they are short DNA sequences that match up with the viral DNA that have previously invaded the bacteria. So when a known viral DNA enters a bacteria cell, there are CRISPR-associated genes that produce Cas proteins, which contain helicases that are responsible for the unwinding of DNA, as well as nucleases that are responsible for the cutting of the DNA. So instead of the virus being able to take control of the cell's machinery, Cas proteins, as well as CRISPR RNA that are associated with the Cas proteins, are produced. Together, they help break down the known viral DNA. Those that match up with CRISPR RNA will be destroyed before an infection can even start. If the DNA of an unidentified virus enters the cell, meaning that there are no spacer DNAs already encoded with viral DNA, then a class 1 Cas protein is produced by Cas genes that is able to break down the viral DNA, as well as taking part of the viral DNA sequence to be copied and inserted into the bacteria's spacer DNA. With the virus's spacer joined with the bacteria's, the new CRISPR sequence is transcribed to produce CRISPR RNAs, which guide bacterial molecular machinery that will destroy any future invading viral genomes. The spacer DNA acts as like a memory system storage for these bacteria. Two female scientists did a study involving the CRISPR system and discovered that the CRISPR-Cas9 system acts as a cut and paste method that could be used to help control or even alter genes. The specific bacteria that these scientists were working with only contained one type of Cas protein, Cas9, which acts as a pair of scissors to cut DNA in two places due to the presence of nucleases. This bacteria's CRISPR system was composed of the Cas9 protein, CRISPR RNA that would match with the viral DNA, and tracer RNA. The way that they altered this bacteria's CRISPR system was by modifying the two RNAs so that they are joined together, forming a single guide RNA. By including the target DNA sequences in single guide RNA, it matches up with the DNA and cuts it according to the new single guide RNA, resulting in deletions, insertions, inversions, or even translocations, all which cause mutations. One example of when CRISPR was successful was when the scientists eliminated the heart defect, hypertrophic cardiomyopathy, within human embryos. These embryos were fertilized in vitro using sperm from males that had a defective MYBPC3 gene, which causes thickening of the heart muscle, which can result in sudden death. By using the CRISPR-Cas9 system, the sequence that composed of the defective MYBPC3 sequence was cut out and replaced with a non-defective MYBPC3 sequence that was encoded by the single guide RNA. Another example of the way CRISPR can help humans is the use of the cell-free CRISPR CPF1 system to improve the speed of cancer diagnosis. Instead of using the Cas protein inside of a cell, a CPF1 enzyme was used to cut the DNA after it was removed from the cell in the form of plasmids. CPF1 was used to cut the DNA because it produced sticky ends rather than blunt ends. What was significant about this study was that it gave scientists the ability to have an exact copy of the mutated tumor sequences so that they can experiment with different types of treatments. The cell-free Crispr CPF1 system also makes it possible to edit more than one gene at a time, which is needed if diseases like Alzheimer's are able to be treated since they involve multiple mutations that need to be replaced.